Hey, it's Mr. Shrum, and we were just about to watch this video, so I'll turn on the captions, and hopefully we won't get a copyright strike. So let's see what happens.
The theory of evolution states that organisms inherit genetic traits over successive generations. Organisms whose traits adapt to the environment survive long enough to pass those traits on to offspring. Species that do not have traits suited for a changing environment may die out and go extinct. Why does that matter? Well, it's believed that all organisms on Earth evolved from a universal common ancestor, and there is plenty of evidence to support this idea. For example, there are 5 major types of vertebrates: mammals, birds, fish, amphibians, and reptiles. These vertebrates are different from each other as adults, but they are curiously similar in the early stages of development. All vertebrate embryos form 3 germ layers: the ectoderm, or outer layer, the mesoderm, or middle layer, and the endoderm, or inner layer. These 3 layers form stem cells inside the blastocyst during the early stages of embryonic development. The stem cells grow into all the different cells in your body. Think about it. All the cells of your body have one type of ancestor cell, the stem cell. All of your cells grew from just one fertilized cell. That's true for all vertebrate life on Earth. Another similarity between vertebrate embryos is that they all look alike. In fact, at this stage, it takes a trained eye to see the difference between a mammal embryo, a reptile embryo, or a fish embryo. And speaking of stages, during development, vertebrate embryos go through a lot of the same stages. For example, vertebrate embryos form pharyngeal arches from the germ layers. However, these arches develop into different organs as the embryos mature. In fish embryos, the arches develop into gills. In humans, the arches form the ears, jaw, and internal tissues of the throat. The fact that all vertebrate embryos develop pharyngeal arches is evidence that they all had an evolutionary ancestor in common. The notochord, a long shaft of cartilage that supports the embryo during development, exists in all vertebrate embryos. A few lower vertebrates keep their notochords as adults, but for most vertebrates, the notochord develops into the vertebral column. Yet the fact that all vertebrates have notochords at one time or another demonstrates a shared evolutionary ancestor. The dorsal nerve cord develops in the backs of all vertebrate embryos. It grows into the central nervous system in all vertebrate adults. Finally, all vertebrates, including humans, have postanal tails as embryos. Some vertebrates keep their tails and others do not. So, yes, at some point in time, you had a tail. The embryos of different species become more easily distinguished from each other as they grow, and yet they all maintain striking similarities. These similarities provide evidence that all vertebrates that will live or have ever lived on Earth diverged from a common evolutionary ancestor.
Okay. So humans had tails. That's always a fun fact to tell people. All right, so another important field that contributes to evolutionary evidence is the study of genetic material in organisms, and the genomes of organisms show remarkable similarities at the cellular and molecular levels. Molecular biologists, analyze the genomes of different organisms based on the similarities to find links in the evolutionary chain. And some scientists maintain that there is just one original DNA structure, and all current species evolved from the DNA of the earliest life forms. They believe that the original DNA structure underwent mutation and recombination to form the different genetic material for the different species. All species, including humans, have evidence of ancestral DNA. Humans have certain genes that are found in some of the earliest prokaryotes on Earth. And comparing such similarities and differences between the genomes of different organisms helps scientists study the possible evolutionary link between them. This comparison helps scientists better understand the process of evolution. All right, so humans and mice have similar genomes. In fact, researchers have found thousands of genes that exist in both mice and humans. What can you conclude about the evolutionary relationship between mice and humans? Do you think these genetic similarities explain why mice are important test animals for human medical research? They don't ask whether that's ethical or not, but oh well, okay. The similarities suggest that humans and mice share a close evolutionary relationship. This makes sense because they are both vertebrates and they are also both mammals. This genetic connection makes mice important research animals because they likely have a similar anatomical structure and body functions. Also, mice could be used to test the effectiveness of different treatments for genetic conditions. They could also be used to evaluate the effect of certain gene mutations on overall health. So, but then you always run into ethical dilemmas. Science can't tell you what to do or whether it's good or bad. That's the only thing holding science back. But now we're going to look at careers in science. An evolutionary geneticist. Evolutionary genetics is a fairly recent field. It has helped us discover evolutionary links between organisms that we wouldn't have known by comparing only their anatomy and development. These geneticists peer into the genetic code of organisms to find similarities between genes. They try to piece together a history of how the genomes of organisms evolved over time. The history of evolutionary genetics. Evolutionary genetics is a field that focuses on the study of genetic variations within a particular species and the effect of this variation in introducing evolutionary changes. Scientists in this field study the genome, different mutations that occur in the genome, changes that lead to speciation, and the different adaptations observed in organisms. Darwin understood that traits were inherited by offspring from parents, and beneficial traits had more probability of prevailing in future generations. However, he had a tough time convincing other scientists of his time. And that's really a staple of all scientists. They find it hard to convince other people of their findings. It wasn't until nearly 100 years later that the work of Charles Darwin was related to classical genetics. So here we have different colors in corn kernels, and this is due to mutations in the genes. You have bluish, blackish corn, red corn, yellow and black corn, red and black. Interesting, very interesting. Now, Darwin's theory was supported and proved by Gregor Mendel. Proved. Proved is a weird word to use here. We'll say supported. Darwin's theory was heavily supported by Gregor Mendel. And Mendel is regarded as the father of modern genetics. And this is because he performed a series of experiments with pea plants, Mendel and his pea plants. And he deduced the importance of genes, alleles, and traits. He proposed 3 laws of inheritance that concluded traits are inherited by offspring from their parents. He also explained segregation and the expression of the traits in organisms. So in this picture here, you have two parent pea plants, green and yellow. This is their offspring, and this is their offspring. So here we'll explain a little more here. DNA sequencing, yeah. The modern geneticists can sequence DNA to find evolutionary connections between organisms. As noted earlier, humans and chimpanzees share almost 99% of DNA, which indicates that we share an evolutionary relationship with chimpanzees. DNA can be analyzed using different methods. Sequencing DNA means analyzing and establishing the order of the four bases that make up the DNA molecule. The sequence tells scientists the kind of genetic information that is carried in a particular DNA segment. This information can be used to establish evolutionary links between organisms. Here, should I suggest a movie? There's a movie, like a science fiction movie based on DNA, and it's called Gattaca. And the title of the movie spells out the letters in the DNA, so GATC. But it's cool. It's like about the future where people with superior genes are like an upper class, and people with lower genes or genetic mutations, they're seen as lower class, and they can't do as many things as the people with superior genes. So check that out. And now we're looking at the Human Genome Project. This was an initiative to understand the DNA sequence in humans and to identify the genes that make up our DNA. The project began in 1990 and took 13 years to complete. Based on data collected, scientists estimate humans have about 20,500 genes. The project also provided information on the structure, organization, and function of the complete set of human genes. The regions of DNA that encoded for genes were sequenced and analyzed, paving the way for advancements in genetics. Evolutionary geneticists can determine evolutionary relationships between different organisms by comparing DNA sequences. Similarities in DNA may indicate a common ancestor. The differences might hint at mutations that enable the organisms to survive in their habitats. Geneticists can also trace when these mutations occurred. Knowing the time period enables geneticists to estimate the development of new traits in organisms and the formation of new species. Geneticists can also trace mutations between populations of a particular species located in different geographical areas. This knowledge helps geneticists determine the places of origin for the species. It also helps that they can determine how changes in specific environments caused each population to evolve over time. So here we're given an article about the study of Galapagos cormorants, and let's check that out. So this is a short-winged Galapagos cormorant and great cormorants on the right, which have larger wings. And all other species have a wingspan meant for flying. So these guys have wings, but they're not meant for flying. Galapagos cormorants are the only flightless cormorant species. They're too small, the wings are too small to lift their heavy bodies. So, in order to trace the genetic changes responsible for these shrunken wings, Alejandro Berga needed DNA from the grounded bird and from a few related species. And this took a year to do. So once these guys split off from their other cormorant brethren, their wings shrunk to 19 centimeters long, and their bodies grew to 3.6 kilograms. So that combination is not flying friendly. Berga suspected he would have difficulty getting permission to collect DNA from the endangered birds. So he emailed anybody who has ever published anything on cormorants in the last 20 years, he says. He found disease ecologist Patricia Parker of the University of Missouri, St. Louis, who had collected blood from Galapagos cormorants in 2000 to monitor the spread of pathogens. So getting to the islands takes special permission, long flights, and boat trips. But getting DNA from the meter-tall birds wasn't hard. They're sluggish, and they just sit there and look at you, Parker says. She shared DNA that had been sitting in her lab refrigerator for more than a decade. Berga used it to reconstruct the cormorant's genetic instruction book, or genome. So that's a good way to describe a genome, is a genetic instruction book for an organism. So next, he needed DNA from closely related species. So that could be the double-crested cormorant, which is the size of a goose. And that bird is protected under a migratory bird treaty between the US and Canada. Since Berga couldn't just trap one and collect DNA, he got a little creative. So he tried to extract DNA from preserved specimens at the Natural History Museum of LA County, but the genetic material was not usable. The San Diego Zoo sent samples of a too distantly related great cormorant. An international bird rescue facility in LA notified him when someone found a dead cormorant on the beach. Berger rushed over, but the bird was a Brandt's cormorant, and that was also too far removed in the family tree to be of use. One email chain led to Paul Wolf, a U.S. Department of Agriculture Wildlife disease biologist monitoring Newcastle disease virus in double-crested cormorant in Minnesota. So with a special permit, Wolf removed one double-crested cormorant egg from a nest. The egg was at just the right stage of development when the wings were beginning to grow to determine which genes are active during wing development. 2 down, 2 to go. While on Alaska's Middleton Island studying seabird parasites, Andrew Ramey of the U.S. Geological Survey collected 2 eggs from a pelagic cormorants for Berga as well. And then he had enlisted Claudio Verdugo, a molecular epidemiologist at the Universidad Austral de Chile in Valdivia. And this bird samples can't be transported between countries because of fears of disease spread. So Berga sent chemicals and protocols to Verdugo, who took DNA from another species, the neotropic cormorant, and then he sent it to Berga. With DNA from 4 cormorant species in hand, Berga and his newfound friends learned that the Galapagos cormorant's stubby wings result in part from mutations in specific genes that encourage limb growth. Berga is now studying how evolution grounded other birds as well. So that was from 2016. Now we can go back to the questions. What did he mean when the cormorant split off from other cormorants? Well, the author meant that the Galapagos cormorants and the mainland cormorants shared a common ancestor. But at some point in the evolutionary history, the Galapagos cormorants evolved into their own species. Do you think the wings are vestigial structures? Yes, they are vestigial structures because a vestigial structures is a part of the body that still exists in an organism, but it does not have its intended function. The cormorants evolved from other species of cormorants that could fly, and a genetic mutation caused their wings to be flightless. So wings that can't fly sounds like a vestigial structure. Why was the genetic information from the distantly related cormorant unusable for this research? Why was it important that they found closely related cormorant species? Species that are closely related will have more similarities in their genetic code than species that are distantly related. The close relationship can help evolutionary scientists pick out the specific changes that occurred in the genetic code to give these birds flightless wings. Are there any similarities between this Galapagos research and Darwin's research? Explain your answer. Well, this modern day researcher used some of the same theories that Darwin proposed. Like Darwin and his finches and tortoises, this scientist understood that the Galapagos cormorants inherited flightless wings. Darwin eventually discovered that his Galapagos finches likely evolved from other species of finches on the mainland. This evolution was similar to how the flightless Galapagos cormorants evolved from other species of cormorants. What modern advancements, or how have modern advancements allowed this modern day scientist to do research in a way that Darwin could not do? So, unlike modern day geneticists, evolutionary geneticists, Darwin did not have the knowledge of modern day genetics or the ability to sequence the genome. So Darwin couldn't have determined that the traits of flightless wings was likely caused by a genetic mutation. And that's the summary. That's the end. So congratulations. That was a good one. The process of evolution. Let me know if you have any questions, and I will talk to you guys next time. Best of luck, and I'll see you later.